Hi, my name is Dr. Nathan Kleczewski, field crop pathologist at University of Illinois Champaign. Thank you very much for hanging out with me today. I'm going to talk a little bit about plant pathogenic nematodes. You heard a lot about soybean cyst nematodes, and that's awesome. But there's other nematodes too. There's other plant pathogenic nematodes, and that's what we're going to talk about today. So we're going to learn a little bit about the basics of nematodes. What are they? How do they work? We're going to talk about our major groups of pathogenic nematodes that affect us here in Illinois, and on how do we know if we have a nematode problem. That's something that is the most important things to keep in mind when we're talking about our field and productivity. If you're an agronomist or you're a CCA, something like that, you need to be able to serve your clientele and make sure that you're giving them the best information that you can. So what are nematodes here? Oh, that's my fault. Some gross ones. That's true. There are some gross ones. These are roundworms. Okay, they're very common throughout the world, and they have many different lifestyles. So if you're at a bar or you're having a party and you want to gross out your friends, just go ahead and type in some of these worms and look for images, and you will not be let down. So plant pathogenic nematodes, those are the ones we're interested in, and these are microscopic roundworms, so they're very small. With a few exceptions, you won't be able to see these with your naked eye. There's about 197 general, 4,300 species of plant pathogenic nematodes. That's across all sorts of different plants, not just field crops, but that's only comprising about 7% of all nematodes that we know of. So it's actually a relatively small amount of nematodes are actually pathogenic on plants. That's important to keep in mind when we're talking about managing these, because a lot of these nematodes have beneficial properties to them as well. I already mentioned this, they're small. What do nematodes look like? So this is if you're to sketch it out and blow it up, this is what nematodes can look like. You can have females and males in the population. They don't always exist together. Sometimes they can switch sexes depending on population numbers, ratios, conditions, things like that. The thing to keep in mind is this little part here, the stylet. This is I'm going to talk about this in a bit, but that's an important feature of a plant pathogenic nematode. That's where it's going to be doing most of its business in terms of plant damage and getting nutrients. These also have a bulb, and this is kind of a muscle that provides that pumping action. So you can kind of think of this whole thing here as almost like a turkey baster, right? And this is the bulb that you squeeze things out and sucks things in. I mentioned also there's a female and a male, and you can see all your typical parts there. The males are kind of neat because they have these little things, these little hooks on the end called spicules, and they also might have tails. And what those do is, think of these, they don't have arms or legs or anything. So if you're trying to mate, you want to make sure that you're lining up your parts with the correct parts. And so these are kind of like little hooks that lock them in. And so when nematodes mate, the male will find the female, and they'll detect it and actually use the fins and stuff to kind of slide down and then find the right parts and kind of lock themselves in, and a little mood music there and everything like that. So stylets, I mentioned, the stylets are very important. There's different types of stylets. There's basically two that we're seeing here. This is an odontous stylet, very large, and actually has guide rings because it's so large. It helps to prevent the nematode from stabbing itself with its own stylet. There's also something called an onchostylet, which we see in some of these as well. It's like a little tooth, so it doesn't have the bulb. But the thing to keep in mind is all of our pathogenic nematodes will have a stylet of some sort, but not all nematodes with stylets are plant pathogens. So like I said, only 7% of nematodes are plant pathogens. All of those 7% have stylets, but you're going to have other nematodes that aren't plant pathogens that can also have stylets. That's why it's important when you send nematode samples out that you send them to a reliable, reputable nematode assay service. We have one of those here at the clinic. They're awesome. They've been awesome for years. And so I would say send your samples to the clinic to get assessments if you are doing any kind of nematode work. These will puncture tissues, and they also will inject chemicals into plants or things that we call effectors, and those can help to prevent the plant from recognizing the nematode, but they also can help with the development of some of these structures, such as galls or feeding sites that are needed for some of these nematodes, such as soybean cyst nematodes, for example. They have many different shapes and sizes. So some of them are very large, like the Longidoras, and the needle sting nematode are pretty big. Foliar nematode, this is a Lesion nematode. And then Meloidogyne root knot nematodes and Heterodora are the cyst nematodes. These are sedentary. So these will actually produce, the females actually have a life stage where they kind of swell up here, and then eggs are produced inside and outside their bodies. So the life cycle for all nematodes is the same. They start out as an egg. They molt in the egg a couple of times. That egg hatches. And then you have these second stage juveniles. Those second stage juveniles are going to be the ones that are typically the infective stage and the ones that we're focusing on when we're doing nematode counts. It's also very important when we're talking about thresholds, when those second stage juveniles occur. A typical life cycle for a nematode, we say, is anywhere between 3 and 4 weeks. That's like a good average range, although depending on the nematode and the environment, you can have some play on either end there. So it can be shorter or larger. Nematodes differ in terms of their lifestyles. So some of them are going to be feeding on the outside of the tissues. That's ectoparasitic versus the inside of tissues. Those are endoparasitic. Those that feed inside the tissues tend to be more damaging because you can think of that nematode moving in that tissue, and then it's going to be causing direct damage to the xylem and phloem and really disrupting the structure and the ability of that root to take up nutrients and water. Ectoparasites oftentimes are feeding on the outside and aren't as damaging. And a lot of the nematodes that we're dealing with in some of crops here, like wheat and soybean and corn, are ectoparasites. And some of the more important ones are going to have that endoparasitic type lifestyle to them. They can move around as juveniles, at vermiform stage where they're little worms here. Those are called migratory. They don't move very far in a year. So you're talking about them maybe moving a foot, 18 inches or so a year. But they can move in soil, limited distances, versus sedentary nematodes. So sedentary nematodes, usually one of the nematodes, that's the female, sets up shop on the root, and then they form specialized structures. You also have some that are migratory and endoparasitic. So these are ones that can move both vertically, horizontally in the soil. They nibble on the outside and the inside of the tissues. These are the ones that are really damaging because these are going in and out, moving around. And you can imagine the more damage to that root, the nematodes causing a lot of that damage, but then you can have secondary invasion by other organisms in the soil. Potentially pathogenic fungi, bacteria, things like that can cause root rot. So typically we're going to be seeing our nematodes in the upper part of the soil, the upper 10 inches of the soil. Some of them can move down in the deeper profiles of the soil, especially to avoid conditions that they might not like, especially things like needle, sting nematodes. They tend to like it cooler and humid, and then when it gets hot, they'll actually go down into the soil deep. So the concentrations, because they're feeding on root, is going to be greatest near the root zone. They are stimulated by root exudates, so sugars, amino acids, secondary metabolites, carbon dioxide produced from respiration in those roots. There's more of that near the plant root, and as you get further away, the concentrations get more dilute, so those nematodes can sense that, and that helps them find the roots. It also helps stimulate egg hatch. As I mentioned, they move, I say a few meters a year, but that's a pretty large estimate. But like I said, 18 inches or so, I think is a pretty reasonable amount to expect it to move in a season. But if you have sandier soils, they can move further. And some nematodes, especially the larger ones, need really sandy soils to cause damage. And so we do see in some of the areas in the river bottoms and areas like that, really sandy soils, you'll start to see some of these things like, sting needle nematode. They can be vectored. That means they can be moved by other animals to other locations or other other organisms to other locations, and they can also vector things as well, especially viruses. So Ziffinema is known to actually transfer some soybean viruses, such as soybean severe stunt virus. But most of the nematodes that we have in our field soils are beneficial, okay? They're just a very small amount that we're dealing with that cause damage. And really it's a rare occasion where we do see them cause damage in our fields. So what are some things that favor nematode development and building up to populations that can cause significant yield losses? Growing monoculture, so the same crop year after year, is going to favor the development of pathogenic nematodes that like to feed on that particular crop. Coarse soils, sandy soils, typically warm, dry conditions are favorable for the development of or the reproduction and movement of nematodes in soils, although some nematodes, like I said, they tend to burrow deeper into the soils and avoid those conditions. And stressed plants. So plants that are leaking exudates are going to be more attractive to these plant pathogenic nematodes than ones that are happy and healthy. The other thing to keep in mind, when you have a healthy plant, the root systems are larger and the plant can tolerate infections much better. So especially in our field crops, they can tolerate a pretty good slug of nematodes before you're going to see significant losses. So general symptoms, what are we looking for in the fields? We're going to be seeing in severely infected fields, plant stunting. You're going to see chlorosis. That means that the tissues typically will become yellow. And oftentimes, again, these are when we have a very susceptible plant and very high levels of the nematode. Those nematodes, if they are causing significant damage, that plant isn't going to be able to utilize its roots to pull nutrients from the soil. And oftentimes that's expressed as a limitation in nitrogen. So you might think you have a nitrogen deficiency, and it would actually show that in a tissue test. But if you're not looking at the soil and what your populations are, you're actually not taking care of the problem, right? You're just, you could potentially be making things worse if you continue to use the same management practices year after year. Those nematodes will continue to build up. So look for yellowing. Typically that chlorosis is going to be very patchy in the field, oftentimes associated with certain areas of the field where it might be sandier or a little drier. In severe cases, the plants can be dead, as you saw in a couple of those earlier images, and you're going to have reduced yields. And I think a lot of times those reduced yields, when you think you've done everything right in your fields, and you don't see any of these other symptoms, you could still have nematode issues that could potentially be reducing your yield. So it might be that you had a more tolerant plant, or maybe the populations weren't quite at that super damaging level, but they were significant enough to reduce yields. So field level, this is what you're seeing. Again, stunted plants, specific parts of the field. And the way I would look at this, if I was in this cornfield, is if I would want to see what the issues were, is I would take samples from within these rows in these hot spots, and I'd also grab some samples from outside, so you have a comparison. I always like to make comparisons so you can see what the differences are in those areas of the field, and that can help you make sure that it's actually a nematode related issue versus not. This is that kind of chlorosis or nutrient deficiency type symptom you might see. But the main point is all plant pathogenic nematodes can have similar effects on the plant, right? And a lot of times they are going to show up as nutrient deficiency and potentially can look like other problems. And so that's why it's important that we keep nematodes in mind. And then I'll talk about later, we sample and we have a plan for nematode management if we need to have them, if we need to manage them. So here's a nice, I could tell you what this nematode is, but you'd be bored, but here's that stylet, remember? So the first one we'll talk about, root knot nematodes. These can be found in soybean and corn. Southern and northern root knot are the most common, though there are other species that you can find. Typically, root knot nematode, southern root knot nematode can infect corn, not necessarily the northern root knot nematode. And we do have southern root knot issues in some fields in Illinois. They are sedentary, so they hatch from eggs. Then the females get on in the root here. Their effectors will cause the production of this giant cell and as gall. And so that's a feeding site for the female that will just deliver nutrients right to that female and not share it with the plant. And then those galls will just continue to grow and swell. And you can imagine if you get hundreds and hundreds of those females on the root over time, you get these really knotty roots. That's not a good thing for the plant and won't be able to take up nutrients very well or water. They also have a very large host range, which makes management of them kind of difficult. The more specific a pathogen or nematode is in terms of its host, the better options we have, and the more options we have in terms of management. It's a little easier. Now you can rotate, although rotation isn't going to provide a ton of benefit because, like I said, a lot of these root knot nematodes will be able to be hosted by other crops and weeds and things like that. We do say typically rotating into soybeans can help, or small grains can help. So root knot isn't going to be infecting roots if the temperatures are less than 65 degrees. That's when we have those small grains in there. Those small grains might be able to promote other nematodes that could potentially feed on some of these root knot nematodes as well as compete with them. Alfalfa is a fairly good rotational crop, oats as well. Again, not very popular, but if you are in an area where you can plant those can help if this is a problem. In the soybeans, you can select resistant varieties, especially those in the warmer climates. So the further south you get, the more of these you can have. So my typical suggestion, if you do have a southern root knot issue in the southern part of the state, is you want to rotate to corn the next year. And then next time you come into that field, select a root knot nematode resistant soybean cultivar. And we've seen success with that before. That would be my recommendation. Anything you can do to increase soil organic matter content. A lot of these are going to be similar for a lot of the different species. Anything you can do to improve soil health or organic matter will increase the diversity of the microbes and other things in the soil that can compete with the pathogenic nematodes for resources and potentially feed on the nematodes and or their overwintering structures like cysts and eggs and things like that. Soybean cyst nematode, and you guys have heard about this for a long time, and we're all very familiar probably with it. I'll bring it up in case you aren't familiar. This is present in 85% of all the fields we have in Illinois. It's our #1 pathogen of soybean, very specific in terms of its host range. So this one isn't going to be infecting corn and small grains. It's going to be focused on our legumes. And it's doing really well on our previously resistant soybean lines. So any of those, the most common resistance we have in our lines. And again, Dr. Greg Tilka at Iowa State has an awesome slide looking at all the available lines of resistance to SCN. And it's like 98, 99% of the available lines are going to be P8788 source of resistance. And then, you know, the rest are other sources of resistance like pecking, for example. So in Illinois, especially in the middle part, northern part of the state, we don't have a lot of resistance options. Though I have heard there's a company that's going to be coming out with some new sources of resistance pretty soon. And that's going to be great if it works well. So if possible, and if I'm talking about managing soybean cyst nematode, I guess I should have this as my second point, but first, if you're in a monoculture and you don't want to rotate to corn or something else, rotate at least your cultivars, because your cultivars will have different levels of the gene that causes them to be more resistant, that resistance to be more effective, even if it's P8788. So if you have something that is doing poorly one year, get a whole different cultivar the next time you go into that field. That can help with reducing your nematode populations. And then, you choose a different source altogether, like Peking or Peking. I'm always Peking. that's, I don't know. When I order duck, I always say Peking. So I guess it's probably Peking. Peking, yeah, it sounds good. I'm gonna have to get that for lunch. Rotating to corn for 1 year can be a really effective way of reducing your nematode populations, your SCN populations. So you have eggs and juveniles in the soil that can overwinter. Some of those eggs will hatch regardless of what crop is in the field. So they'll hatch whether or not soybeans in there or not. It's just a survival strategy. And you might also have some adults and things like that. So if you go into corn, you won't have a host. They won't be able to feed. Those eggs will hatch. Those juveniles will die. Those adults will die, and it can reduce your SCN populations by up to 50%. It's not guaranteed to do that, but it definitely can help reduce those numbers. Okay. Up. That's supposed to be up, not you. U2. Needle nematode in corn, it's huge. It's a big one. This is something that we see in our very sandy soils, typically going to be in areas that are irrigated. And again, those are where we're irrigating our sandy soils. River bottoms here in Illinois are our biggest problem. And the thing is, when you irrigate, it actually can make these issues worse because these nematodes, needle nematodes, like that condition when it's cool and moist. And so when it gets hot and dry, they can move several feet into the profile. And that can actually, those dry conditions can allow the plants to recover and, set out new roots. And, these needle nematodes won't be an issue because these are so big and they cause a large amount of physical damage to the roots. And so really damaging a single nematode is a threshold for needle nematode, I say. But because of this, them moving down into the soil, when you sample, can miss a lot of these guys. So sampling in the middle of the season isn't a good way to get needle nematodes. You're going to want to sample earlier in the season to see if you have needle nematode issues, most likely, if it's hot and dry. Rotation for at least a year to a legume such as soybeans and alfalfa can be very beneficial. And this is something I should have mentioned for the other nematodes too. Weed management is essential. Good burn down at least a couple weeks before you get into your field and plant, take away potential food sources. So a lot of these nematodes, they can feed on some of these weeds that we have growing in our fields. But that's pretty much how you're going to be managing needle nematode. If you're on sandy ground with a history of needle nematode, your irrigation management is going to be important as well. Minimizing your irrigation early in the season and maybe focusing on more frequent but less amounts after you start getting the tassels coming out. That's probably when you're going to really need that irrigation if it is dry. Lesion nematodes in soybean and corn can potentially be problematic, but there's a lot of different species in Illinois. I want to see there's somewhere between 7 and 9 different species. I don't know, and it's kind of up in the air. I need to look and see what else we've found in some of our other vegetable crops and other things like that. The one that's best studied is this Pathogenic penetrans. When you do assays and when we're doing assays of lesion nematodes for clinics and things like that, they're not speciated. They're just called Pathogenic, and it's very possible, and some speculate that these different species cause different amounts of damage or potential damage to the plant. And so, the damage thresholds, those are always kind of a tough thing in nematodes, but even harder with the lesion nematodes because, we don't know what's out there. They could have mixed populations and, just hard to figure out, but they do hatch from eggs. And then what they'll do is they'll feed inside the roots and cause a lot of damage. So you can imagine that's going to cause those roots to get this, that's why they're called lesion nematodes, develop these kind of black rotted things. A lot of secondary organisms get into those roots. You can get root rots associated with these as well. Usually like it, when the soil's warm, like most of these nematodes. So it needs to be over 68 degrees Fahrenheit before you really see these guys get active out in the soil. In terms of managing lesion nematodes, because they do have a wide host range and there's so many species out there, rotation isn't really a great option, but there are some poor hosts like alfalfa and wheat are usually poor hosts. Again, burn down and anything you can do to reduce plant stress will help with these. Sting nematodes, something that we typically see in sod production rotated with corn. And you can see the type of damage that can be caused by these sting nematodes here, really reducing and nipping the meristems here on these root tissues or the root systems, and that's going to impact those plants significantly. Very sandy soils, so you're going to see these again, much like needle nematodes in those areas where it's really sandy, or like I said, sod production, but you're growing sod on sandy areas anyway. And in those areas where you do see it significant at significant levels, it can cause complete yield loss. And this is a really big issue in turf grass production, golf courses, sod, that sort of thing. You do not want to get sting nematode in your turf grass. There's a lot of other nematodes as well, and a lot of these will be detected if you send in a sample to a clinic or a service. A lot of these by themselves, I'd say most of these, I'd say all of them, let's say all of these, have very questionable thresholds, and also if they have a questionable threshold, it's also not likely, we don't likely know what sort of damage they have. We just know they can feed on the roots. A lot of times the plants tolerate these very well. We don't see a yield response very reliably when these nematodes are the ones that are in high abundance. We did a corn nematode survey in 2018, 2020. And so what we did is we went out and looked at fields that were planted in corn. We sampled these between V5 and V8. That's so that we're able to, one, we have root systems. Two, the fields are warm, but not necessarily hot. And again, we don't want to miss nematodes such as needle nematode or sting nematode, right? We sample within the rows, not in between rows. That's how you want to take these samples, making sure we get 8 inches deep or so, getting that root material. And then these are processed by Naturator's lab using a couple of standard techniques. These were the counties that were assessed in 2018. There are at least 2 fields 10 miles apart within each county. These are randomly picked fields, so we're not trying to pick fields that have a history of nematode problems. We're just trying to get an idea of what's likely to be encountered in our fields in Illinois. And here's where we had 2020s. You can see we got some new fields in 2020, a couple that we missed in 2018. Didn't do it in 2019 because you remember all that rain we had. Field growth stages were all over the place. It just wasn't a good year to do a survey. Plus it was wet. So if we look at our 2018 samples, spiral and lesion have the highest frequency of fields with them present. We also see that a lot of our fields still have SCN juveniles. Remember I said that's why having rotating corn is one way to reduce those numbers. So we still had about 65, 66% of our fields had soybean cyst nematodes in them, and those are the corn fields. We did see one field with needle nematode. Didn't see, interestingly enough, in our corn, didn't see any root knot, didn't see any sting in this. And again, we're limited by the number of samples we can take as well as the number of counties. So I'm not saying we don't have those, but we didn't detect them in our 2018 surveys. In 2020, it's a similar thing again. We're seeing spiral and lesion and SCN, again, at relatively similar frequencies and a little bit of needle, but again, no root knot nematode, nothing, no sting nematode, but we do get a lot of these other guys. And the thing to keep in mind too is a lot of these nematodes are together in our fields. So it's not like it's just a field and they only have lesion. It's going to have lesion and spiral and, lance or even more. And so there's always this question of the accumulation of samples or accumulation of nematodes in a sample and what that means. for yield impacts. But the thing to keep in mind here, our lesion nematode, the one that I would say is potentially most concerning in corn, well, not most concerning, but might be a problem, is fairly frequent. But the number of samples that were what we would say over threshold, very, very few samples. So the idea that you have to be managing nematodes on every acre of corn and there's a 10 bushel yield loss is probably only true in those fields where you have severe levels of some of these organisms. And I'm just pointing out that there's a couple other nematodes here that we have some that are exceeding thresholds, but very few. And our overall is about 10% of the fields we sampled. Okay. So spiral lesion SCN, 3 or more in all samples. We don't know the cumulative effects. Didn't see many needles, but again, it's a limited survey. Same with root knot nematode and sting. The thing to keep in mind is that at least in corn, if you're just a regular grower, the likelihood that you're seeing a significant impact of nematodes on your yields is fairly low. That doesn't mean you shouldn't pay attention to them, just means that it's unlikely that you need to do something to manage them right now. I say sample every 3 to 5 years. They vary in time and space, so sampling them can be very, very challenging, and you have to do it correctly or you're not going to get good results. So you need to make sure if you're going to sample, you have a cooler with you, plastic bags, ice in that cooler, a probe or a shovel, markers to take down information. You need to know where you're going to send it and send it as soon as possible. I tell people, take your samples on a Monday through Wednesday and send them out so that they get to that clinic on a Thursday or Friday. You can keep these in a cooler on ice, or you can keep them in a refrigerator for a couple of weeks, up to a month, but you just got to be cognizant that the faster you get them in, the cooler the samples stay and the fresher they are, the better your results are going to be. I like to sample corn early, V6 to V8 for just a look-see to see what's in there. And we want to make sure that our soils are loose, get in between the roots, go in at a 45-degree angle if you have a probe, and make sure you're getting the root fragments in there as well. And you're going to be sampling in a zigzag pattern, usually about 15 cores for every 10 acres. So you want to make sure you're sectioning your field into 2 quadrants or something like that, 10-acre quadrants or 10-acre sections. Mix those together, and then you want to send those out. With soybean, we're focused mostly on our soybean cyst nematode. So typically we want to sample those after harvest, but you can sample in the spring as well. They'll still be there. But if you sample in the fall when you do your nutrients, that gives the clinic plenty of time to process them, and you have plenty of time to get those numbers before you put something in that field. Again, same kind of sampling strategy. And sending those out. And I say send them out if you can. I like to use the UIUC service. They're very good. And the one thing that you will get from them is really good quality data and reliable data. What you don't want to do is change where you're sending your samples. So always go to the same source year after year, because different labs will give you sometimes different results, but they're typically consistent within a lab. So base your management practices off the recommendations from a single lab. Don't mix them up. But check it out. Send them in different labs. See what the numbers come back as. It'd be an interesting exercise. So things to keep in mind here. Pathogenic nematodes are present at some level in all of your fields, but not often yield limiting. And we talk about in corn, we only saw 10% of our fields with a nematode with one nematode over what would be considered to be a threshold level. SCN is our biggest problem in soybeans. We're going to be really concerned if we have needle and sting in our corn, and root knot can be problematic in the southern parts. Sampling is going to be essential. And you want to make sure, like I said, you're sampling every 3 to 5 years in all your fields, and you're keeping track of your records so you can see how your practices are doing, and if you need to implement some nematode management strategies if something appears to be building in abundance over time. So thank you very much. Have a great day, and I will talk to you later.